Hi guys, so today's video is all about microscopes and magnification. So if you want to know what a microscope is, how it works, and how light microscopes are different to electron microscopes, this is the video for you. I'm also going to include some worked examples of magnification to really help you nail this topic, and we'll even go through some standard form at the end of the video too. So stay tuned guys, and like, comment, subscribe. Let's get into the video. So welcome to GCSE Science Biology Revision, Microscopes and Magnification. I'm going to take you all the way through microscopes and magnification and make this interesting and fascinating topic simple for you guys to understand and ace in the exams. So what are microscopes first of all then? Well, microscopes allow us to see the structure of cells and organelles. Magnification is the number of times bigger an image is than the object. Resolution is the minimum distance between 2 points that can be distinguished between, so you can see finer details. So higher resolution microscopes allow you to see more detail and more structure. Light microscopes next of all. Now, light microscopes magnify an image of a specimen using light and lenses. The specimen needs to be thin so the light can pass through. For some specimens, you just want them to be one cell thick so you can get lots of light through and really clearly see your image. Now, light microscopes allow us to view living specimens, cells, and even larger organelles like nuclei. And organelles are like the small structures within cells, such as the nucleus or the mitochondria, or even like the chloroplasts in plant cells. Now, stains can be applied to specimens to enable us to see certain details. Now, light microscopes, you need to know the structure of the light microscope. So let's go through and label up this nice image of a light microscope here. So this part here is the eyepiece. Now this microscope has a binocular lens, meaning it's got two eyepieces, but you could just have a single one. Next of all, we have the lenses, the objective lenses. Now they're the ones that can be twizzled round so you can select the power of lens you want. We have the stage there that we place the specimen on. We have the light source that will shine the light through our thin specimens to enable us to see the details. Here we have the arm. Then we have the coarse focusing knob, the fine focusing knob, and then the base at the bottom there. So I highly recommend drawing a diagram and labeling it up as part of your revision. Then you could even cover it up and see if you can remember the parts. So next of all, electron microscopes. Well, electron microscopes use a beam of electrons instead of light. So that's a really key difference, and it's a big reason that electron microscopes have a higher resolution. Now, like I've just said, they have a greater resolution, but they also have a greater magnification than light microscopes. Electron microscopes are used to view the detailed structure of cells and organelles like chloroplasts, which we've already mentioned are in plant cells. So how is magnification calculated then? Well, magnification equals image size divided by actual size. So that's an equation you need to memorize. Now, the golden rule before you do any magnification calculations is to make sure you've converted your units first. So if the question asks for micrometers, make sure you calculate any measurements or convert any measurements into micrometers before you get into the equations. So let's have a look at the unit conversions. Well, 1 millimeter is 1,000 micrometers. So to go from millimeters to micrometers, you times by 1,000. And 1 micrometer is 0.001 millimeters. So to go from micrometers to millimeters, you divide by 1,000, because obviously millimeters are 1,000 times bigger than micrometers. Now here we've got the triangle, and I always say to my students, remember, think to yourself in that exam, I am a pro at calculating magnification. So the I stands for image size, and the am is actual size and magnification. Now, you can remember I over AM, I am, okay?
And all you simply do is cover up the part you're trying to work out. So for example, if you cover up image size, you do actual size times magnification. If you cover up actual size, you'll do image size divided by magnification. And if you cover up magnification, you'll do image size divided by actual size. So let's go through some examples now, because I always find doing a practice question is the best way to learn this. So question 1, a chloroplast is 8 micrometers in length. The image size is 15 millimeters. What is the magnification? So pause the video here and I'll go through the answer. So let's have a look at how to get the answer then. So the first thing to do is to convert your units. You're given the image size in millimeters, so we've got to convert that to micrometers. So all we simply do is times it by 1,000. So 15 millimeters equals 15,000 micrometers. So we've made it the same as what the chloroplast is measured in. Then we do our equation, so magnification equals image divided by actual, or in this case, 15,000, which is our measurement, divided by 8, which is our chloroplast length, our actual size, and that gives us 1,875 times magnification. Did you get that one right, guys? So let's have another example. This is our question. An image of a chloroplast is 16 millimeters. The magnification is 3,000 times. What is the actual size in micrometers? Pause the video here and then I'll go through the answer. So let's have a look at how we work it out then. So convert your units. So the image will be 16,000 micrometers and the actual size will equal the image divided by the magnification, remembering to cover up on the triangle. So that'll be 16,000, which is the image of the chloroplast, divided by 3,000, which is the magnification, and that will give us 5.33 micrometers. So let's have a little look at standard form next of all then. Well, standard form is a great way of making large numbers easier to work with. So 0.0005 is 5 times 10 to the minus 4. Now, it's okay, people like myself saying it's easy to work with, but you've got to get the hang of it first. Once you've got the hang of it, it is 100% easier to work with, but to begin with, it can be a little bit tricky. Now, 5 million, or 5 with 6 zeros after it, would be 5 times 10 to the 6. And I always tell my students, just count the zeros. So if it's 5 million, it's got 6 zeros, take away the zeros is 5, and then it'd be 10 to the power of 6 because there's 6 zeros, because you move the decimal place 1, 2, 3, 4, 5, 6 places. Now, in the top example, we move the decimal place 4 places, so we move it 1, 2, 3, 4 to give us 5.0. So it'll be 5, which is the same as 5.0 times 10 to the minus 4. And basically, it's just the direction you move the decimal point is whether it's a positive, 10 to the power of, or a negative. So the number of places the decimal place moves is represented by the power of 10. So here's a question next of all, guys. What would 56 billion, or 56 with 9 zeros, be in standard form? Pause the video, write down your answer, and we'll go through it. So the answer then, guys, is 5.6 times 10 to the power of 10. Did you get it right? Drop in the comments if you did or not. Don't forget to like, comment, and subscribe, and I will see you in the next one. Good luck with the revision.